Hi, Suzanne.
Hi, Aaron.
Hi, Yukiko. Hey, Suzanne, you're still muted.
Can you hear me now?
Yes, perfect.
Yes,
Thank you.
great.
Aaron, where are you?
I'm in Boston.
Okay,
Just there's some Huntington's disease meeting I'm attending.
great. Um, Maybe Yukiko wants to show her slides first.
Yeah,
Okay,
yeah, you can both test your slides.
all right. So, my goodness, not this one. No.
Aaron, I heard you gave an amazing talk in Spain.
Oh, wow, thanks. I don't know if it was amazing, but it was a great meeting. Dario was there.
He said he got to know you quite a lot at this meeting. He was quite impressed with some of your secret talents.
Oh, yeah, he had yeah, so he had a bunch of friends with him, and I wanted to go down to the beach into the Mediterranean in the middle of the night. And they said, no, we don't want to go. I said, if I could show you something amazing, will you go? And then they said, okay. So then I showed them and they said, wow, that was amazing. But then they ended up not going anyway.
Well, Dario was impressed with your magician skills.
Yeah. So,
You'll have to show us some tricks sometime.
okay.
So, actually, I'm having a trouble here.
Oh, okay. So,
Oops.
what do you see like the green button that says share screen?
Yeah. But I have dual screen, but
Do,
Oh,
oh,
dual
I
screens.
yeah, and I cannot select the screen actually. I don't know what to do with this, but all right.
uh, you
Can you see it?
dual screen. You
Looks
might,
good.
can you do like just,
If you hit display, it should
yeah,
work.
yeah. And now try,
Oh, now it's
oh, okay, so
presenting us.
go to the thing that says swap displays. Do you see in the top left? There's like whatever. Yeah, there you go. Perfect. Yeah, that
Is it
looks
okay?
Great.
good.
Good.
Yeah, that looks good.
Right. Then okay, I will stop this.
I'll
Yeah.
try mine really quick. Okay, let's see if this is going to work. I have to allow Zoom to share my screen. Hold on.
I was just having dinner with Wade Harper, Suzanne, and he
Oh,
mentioned
yeah.
that you're hosting a bunch of them in December.
December 12th, I'm going to host a cell biology of Parkinson's disease mini conference,
Wow. If
and
you
you
need
are
someone
welcome.
to bring, like, I can carry around the microphone or something, or help
No, you
out
don't need
with.
to carry out the microphone, but I'll tell you more offline. I have to figure
Yeah.
out how to share preferences for Zoom. Um, Security and privacy,
Oh,
maybe?
yeah. To restart it.
Doesn't want to do that.
Um, do you see the security and preferences?
Yeah, I'm getting there. It wouldn't open
Okay.
a second ago.
Okay.
Oh, here we go.
Meanwhile, how's Tokyo, Yukiko?
Yeah, it's been the all of the incidents and the patients of COVID-19 increases a lot, but
Hmm.
the yeah the in general the life comes back,
Hmm.
and so people don't care
It's
about
just,
that so much.
looks okay to you, Aaron?
Yeah,
Looks great. Yeah.
Great.
perfect,
Okay, so I'll unshare.
beautiful.
Perfect. Yukiko, is it the BA5 variant?
Yes, yes. Yeah, it's the dominant species right now. So I got infected last month, but it was very mild. So it was like vaccine.
I talked to Tomomi. She seemed to get like 3 times, or infected like 3 times already.
Already?
Yeah,
Wow. How about you?
once, just once. Yeah,
Mhm.
I got it once.
Uh-huh.
BA5.
that's a good one to get if you have to pick one to get. It seems.
Aaron,
Hi, so long.
hi Yuki. Nice to see you.
Hello.
Hi.
All right. So I'm gonna share the
Hi,
screen.
Hideki.
But before you share, Yukiko, I'm gonna give like some
Oh, okay. Okay.
yeah. Well, I'll share one
Sure.
slide in the beginning.
Mhm.
Sure.
Where are you? You're not in California?
I'm in Boston.
Oh, okay.
Yeah, in a hotel in Boston.
Mhm.
How's Shanghai doing?
Hot, super hot. It's craziest summer all over the world. So today is supposed to be the last hottest day in Shanghai. Tomorrow it's get drop down 5 degrees and seems to be fine.
Okay.
Fingers crossed for you.
Oh,
We have a
yeah.
very mild summer in Stanford. It's actually very pleasant, but nowhere
Yeah,
else.
California is always great.
Yep.
In Europe, I saw it's also very good, crazily hot. Right?
Yeah. It was
Mm.
hot. Hideyuki, are you still in Boston or you're back in Japan?
Now I'm back in Japan the day before
Oh.
yesterday, unfortunately.
Oh, I came to Boston yesterday.
Oh, really?
Yeah. Sorry, I missed you.
Just impressed. Are you Marriott?
I'm in the Marriott. Yeah. Do you
Oh, okay.
recognize the
Yeah.
painting? Everything's good back at Stanford, Suzanne?
Everything's fine. Yeah.
Normally, the undergraduates will be able to come into the classroom starting a normal. Sweet.
Yeah, I think they still have to wear masks in the classroom, but in their living situations, they don't have to so much. And they'll try to stage the return of the students to campus so that everyone is tested and try to get back to normal a little bit.
Hopefully that goes well.
Yeah, I missed the huge class I had once or twice a year in Shanghai, but has been canceled for a while.
Are they going to start that up again? So?
Yeah, I guess so. Yeah, they're coming for they have a few hundred people big class. It's very boring to have a class online. You know?
Yeah.
You don't feel the students at all. You don't know where they're
Right.
at. Yeah, I
Well,
agree.
yeah, but no idea what these.
Exactly. Even though I love listening to these Zoom talks every week, I actually hate giving Zoom talks, ironically.
Yeah.
Then you'll be
It's funny.
sympathetic with us today.
Yes. No, this one is different. This one is a lot of fun, and this will be great. Maybe we'll wait a few seconds and then get started.
I was just going to add a moment ago that Hideyuki gave a great talk at MIT a few weeks ago that I heard, but I was on mute when I tried to say it.
Great.
In person, MIT. Yeah, great.
I was complimenting your talk at MIT a few weeks ago.
Oh,
Okay, maybe Zilong, should we
Sure.
get started?
Sure,
Okay.
go ahead. Yeah.
Okay, hi everyone. Welcome to another week of Neuro Zoom broadcasting today from Boston. And we have two great speakers coming up, but let me advertise next week's talks. We have Zhe Zhang from Institute of Neuroscience in Shanghai and Weiguang Li from Fudan University. So please tune in for those talks and keep contacting Zilong or me if you want to talk about your latest research. We have slots available in the fall, and we'll keep going. And now we'll get started with Yukiko Gotoh, and Zilong will introduce her.
Sure. Okay, it's my great pleasure to introduce Professor Yukiko Gotoh from University of Tokyo. Actually, I became a big fan of Yuki's work since I started to learn neurobiology, neurodevelopment with animal goats in UCSD back almost 16 years ago. So Yuki has did her bachelor degree, master degree, and PhD in University of Tokyo. Now he's professor at Tokyo. So after a PhD, he briefly went to US to work with Jonathan Cooper, Mike Greenberg. So in her own lab in Tokyo, University of Tokyo, Yuki has used a series of elegant assays to study the role of epigenetic modulators, such as polycom in neurodevelopment. So I'm always still amazed by a series of amazing work by her group. So today she will talk about her latest, the embryonic origin of adult subventricular neural stem cells. So welcome, Yuki. Take it away.
Thank you, Zilong.
Okay.
So
Yeah, great.
Is it okay?
Yeah, great.
Right. So first of all, I'd like to thank Aaron and Zilong for organizing this very meaningful online seminar series, which gives everybody in the world an opportunity to get together, even under difficult situations like COVID. So, and thank you for inviting me to this. So I assume that many of the people in this audience are not familiar with my work. So I'd like to start with some introduction of some background and then talk about our recent study. So here I'd like to compare embryonic tissue stem cells and adult tissue stem cells in general. The mission of embryonic tissue stem cells is development of the tissue. By contrast, the mission of adult tissue stem cell is the maintenance of the tissue, as well as the adaptation to environmental changes. In the case of adult neural stem cells, their functions have been studied extensively in mice. Adult neural stem cells reside in two places in the brain, the subventricular zone of the lateral ventricles and subgranule zone of the hippocampal dentate gyrus. So newborn neurons generated at these two places contribute to various cognitive functions such as learning and memory, including forgetting, mood control, innate behaviors, as well as damage repair. So they play very important roles in the adult brain. In mice, adult neurogenesis can be regulated by various extrinsic inputs. For example, dentate gyrus neurogenesis is enhanced by enriched environment, running neuronal activities, injury, and antidepressant. Whereas adult neurogenesis can be decreased by stress or aging. And the defects in adult neurogenesis has been associated with impaired cognitive functions and mood control, both in mice and humans, especially in the context of psychiatric and neurodegenerative diseases. So here, I'd like to emphasize that one remarkable feature of adult stem cells in some tissues is quiescence. They rarely divide. However, when necessary, they start to divide and produce differentiated cells in need and come back to their quiescent state again. Then, why are these adult tissue stem cells kept in quiescence? It's been proposed that quiescence is essential for the long-term maintenance of adult tissue stem cells. In the case of adult neural stem cells, we have previously shown that the CDK inhibitor 57KIP2 is essential for the quiescence. So we found that 57 knockout in the adult neural stem cells reduce quiescent neural stem cells and increase proliferating neural stem cells and newborn neurons. Importantly, we found that the long-term deletion of 57 resulted in the exhaustion of neural stem cells. So 57 knockout in the adult neural stem cells for 2 years reduced the number of neural stem cells, total proliferating cells, and the newborn neurons. So 57 blocks the cell cycle, and 57 knockout accelerates the cell division and enhances neuronal production at the beginning. But eventually, this results in premature exhaustion of neural stem cells. So this study and many other studies in other tissue stem cells, such as hematopoietic stem cells, indicated the essential role of quiescence in the long-term maintenance of adult tissue stem cells. However, it still remains unclear why quiescence is beneficial for the long-term maintenance. So this may be due to limitation of division times, called Hayflick limit, but this is not proven. So I will come back to this point later in this talk. So anyway, here I'd like to emphasize a sharp contrast between embryonic neural stem cells and adult neural stem cells. So embryonic neural stem cells divide rapidly to make a brain within a limited time period, whereas adult neural stem cells are kept in a quiescent state and divide very slowly for their long-term maintenance. So an important question arises. How does the origin of adult neural stem cells emerge from embryonic neural stem cells during development? So in a previous model 10 years ago, adult neural stem cells are thought to be randomly selected from embryonic neural stem cells after brain development. However, this is a bit puzzling because if there is a limit of division times, many rounds of cell division during development may result in exhaustion of adult neural stem cells earlier. Also, by the end of the development, most embryonic neural stem cells stop producing neurons and produce only glial cells, which is called gliogenic phase. However, adult neural stem cells produce neurons. So it doesn't seem natural that gliogenic neural stem cells produce adult neural stem cells. So we hypothesized that the embryonic origin of adult neural stem cells may be distinct from embryonic neural stem cells dedicated to the brain development. And we also hypothesized that if they are distinct, the origin of adult neural stem cells may be slowly dividing, just like adult neural stem cells, for their long-term maintenance. So we perform in vivo H2B-GFP retention analysis to estimate the cell division number. In this analysis, we induce H2B-GFP, so GFP fused to histone H2B transiently by using a TET-on system with a single doxycycline injection. In rapidly dividing cells, H2B-GFP gets diluted quickly and becomes undetectable. But slowly cycling cells are expected to retain high-level H2B-GFP for a longer period, okay? So we injected doxycycline at E9, and after 1 day at E10, almost all the neural stem cells in the ventricular zone were strongly labeled by H2B-GFP quite homogeneously. However, after 6 days of dilution at E15, only a fraction of cells in the ventricular zone retained high-level H2B-GFP. So these H2B-GFP retaining cells were positive for neural stem cell markers such as SOX2. So therefore, slowly dividing neural stem cells indeed exist in the embryonic brain. Remarkably, some of these cells still retain high-level H2B-GFP even at P28 after 38 days of dilution. Importantly, these cells show typical features of adult neural stem cells. So this suggests that a majority of adult neural stem cells at the subventricular zone are derived from slowly dividing embryonic neural stem cells. So these results indeed demonstrated that slowly dividing embryonic neural stem cells are set aside in the early stage of development, and they later become adult neural stem cells in the subventricular zone that produce neurons throughout life. So these lineages, the neural stem cells for development and neural stem cells for adult neurogenesis, are indeed distinct from the beginning. So Alto Alvarez-Buyos group also came to the same conclusion. So this is about the ganglionic eminences, and Fleda Miller's group showed that neural stem cells in the neocortex also slow down the cell cycle during the embryonic stage and become similar to adult neural stem cells. Then a big remaining question is, how are the origin of adult neural stem cells established at the beginning? So we have shown that the CDK inhibitor 57 actually plays a causal role in the establishment of this adult neural stem cell lineage. For example, 57 knockout in the embryonic neural stem cells markedly reduced the number of adult neural stem cells. So these green cells, SOX2 positive cells lining the ventricular surface, are adult neural stem cells and their progeny. And you can see the marked reduction by 57 knockout. So 57 expression appears to play a pivotal role in establishing adult neural stem cells. Then how does P57 work? So Eugene Harada, a talented staff scientist in my laboratory, asked this question. So Eugene overexpressed 57 in rapidly dividing neural stem cells in the neocortex and examined what kind of genes are upregulated by 57. Surprisingly, 57 expression alone was sufficient to make a global transcriptional profile close to adult neural stem cell, adult quiescent neural stem cells in this gene set enrichment analysis. So this is an amazing enrichment. So we indeed found that the 57 expression induces not just the quiescence, but also various features of adult neural stem cells. So here, I'd like to focus on the Notch signaling pathway. So Notch signaling is known to play a central role in the maintenance of both embryonic neural stem cells and adult neural stem cells. As you know, activation of Notch signaling induces expression of Hess and Hay family transcription factors, which in turn suppress proneural genes and thereby maintain the undifferentiated state of neural stem cells. So Eugene actually found that the level of active Notch1 protein was much higher in slowly dividing neural stem cells compared to rapidly dividing neural stem cells. So these H2B-GFP retaining neural stem cells are slowly dividing neural stem cells. And you can see that the level of cleaved Notch1, active Notch1, was higher in these slowly dividing neural stem cells compared to rapidly dividing neural stem cells. Then, is there any difference in the Notch effectors? As I mentioned, the Hess-Hay family transcription factors are the major downstream effectors of NOT signaling, which inhibit proneural genes. In rapidly dividing neural stem cells in the neocortex, HES1 and HES5 play a major role in mediating NOT signaling. So Dr. Kageyama's group has shown that in rapidly dividing proliferating neural stem cells, the expression of HES1 and HES5 oscillates at about 90 minutes to 2-hour cycle based on their negative feedback, by which HES1 and HES5 proteins suppress their own gene promoters. So this oscillatory expression of HES1 and HES5 result in oscillatory and low-level expression of downstream targets such as proneural genes, which enables neural stem cells to balance between maintenance and neuronal differentiation. So HES1 and HES5 are expressed in rapidly dividing neural stem cells. Then what about in slowly dividing embryonic neural stem cells? So interestingly, again, in this H2B-GFP retention analysis, we found that the expression of HEI1 and HEI2 was much higher in slowly dividing neural stem cells compared to rapidly dividing neural stem cells. So unexpectedly, HEI1 and HEI2 were more enriched in slowly dividing neural stem cells compared to rapidly dividing neural stem cells. Then, are there any different properties between Hess and Hay family members? So we found that HEI1 and HEI2 messenger RNA are much more stable than HES1 and HES5 messenger RNA in the neural stem cell cultures. As you can see, the degradation of HEI1 and HEI2 messenger RNA after the addition of actinomycin D was much slower than the degradation of HES1 and HES5 messenger RNA. Actually, this can be a very important difference because the short half-lives are necessary for the oscillatory dynamics of HES1 and HES5 expression. So if their degradation is not fast enough, they cannot oscillate. So HEI1 can also suppress its own promoter through NBOX, but because of its stable nature, we assume that HEI1 may not oscillate. So we perform a time-lapse imaging of a reporter of HEI1 promoter activity in collaboration with Dr. Yamada, Imayoshi, and Kageyama at Kyoto University. And very interestingly, we indeed found that the HEI1 reporter does not oscillate. So his 5 reporter obviously oscillated at about 90-minute cycle, but HEI1 reporter did not appear to oscillate within this time scale. So this finding may have an important implication, because if HEI1 does not oscillate, HEI1 may be able to constantly suppress the differentiation-inducing factors for neural genes such as ACL1, which may result in robust maintenance of the undifferentiated state and long-term maintenance of neural stem cells. And Eugene indeed found that HEI1 knockdown reduced the neural stem cells and increased prematurely differentiated cells positive for ACL1. As a result, HEI1 knockout mice showed a reduced number of postnatal neural stem cells at the dorsolateral ganglionic eminences. So these results support the notion that HEI1 is necessary for the robust maintenance of neural stem cells. So these results indicated that 57-induced cell cycle arrest can activate NOT signaling and induce sustained non-oscillatory HEI1 expression, which may in turn result in robust and long-term maintenance of neural stem cells and contribute to the establishment of adult neural stem cell lineage. Actually, we found that 57-induced cell cycle arrest induces various features of adult neural stem cells in addition to the Notch-Hay signaling, such as enhanced lysosome biogenesis, fatty acid oxidation, and antioxidation pathways, all of which may also play a role in the robust maintenance of neural stem cells. All right, so this is the question that I mentioned before. Why is the quiescence beneficial for the long-term maintenance? The limitation of division times may be one of the reasons, but in addition to it, here I'd like to propose that the quiescence may actively trigger a program for stem cell maintenance. So my take-home message here is that the cell cycle arrest of stem cells is not just about stopping proliferation, but it actively triggers various signaling pathways such as Notch-Hay signaling, which promote adult neural stem cell specific properties. Then you may wonder how cell cycle arrest activates Notch signaling. So one possible explanation is this. So Kim Dale's group has shown that CDK1 and CDK2 phosphorylate cleaved notch, active notch, and this phosphorylation promotes degradation of active notch. So therefore, cell cycle arrest is expected to stabilize active notch. So cell cycle arrest can initiate various events like this. So by the way, you might wonder, what kind of signals may trigger 57 expression in embryonic neural stem cells at the very beginning? We found that 57 is regulated at the protein level rather than the transcription level in the embryonic neural stem cells. And 57 protein is known to be stabilized in response to BMP signaling. So actually, we found that BMP6 starts to be expressed at the dorsolateral ganglionic eminences at around E14 to E15. And also, BMP ligands such as BMP245 are highly expressed at the colloid plexus and secreted into the cerebrospinal fluid. So BMP may be involved in the stabilization of 57 in the embryonic neural stem cells. And interestingly, we found that the treatment of embryonic neural stem cells with BMP2 or BMP6 increased the adult neural stem cell markers such as HEY1 or DLK1. So given the essential role of HEY1 in the establishment of adult neural stem cells, this suggests that BMP signaling plays a role in the emergence of embryonic origin of adult neural stem cells. So we are now examining the role of BMP signaling in the emergence of adult neural stem cell lineage via 57. So these are the people who contribute to this work, especially the Eugene Harada did almost all the work regarding the HEY1 in establishing adult neural stem cell origin. And thank you very much for your attention.
Okay, thank you, Yukiko, for great, beautiful talk. I'll open questions. So may I ask the first quick question? So I'm quite interested in adult neurogenesis recently. So I found out one of our genetic mutations on autism models. There's some gene expression in their different adult neurogenesis since cells around the SVZ. So what was your suggestion to find out whether it has some reactivation or some different things happening in the adult neural stem cells? What should we looking at?
So you're talking about the disease model?
Cells around the SVZ. Yeah. In our one without this model of something happened, gene co-label in the adult neural SVZ. So what's your suggestion? What gene should we look at? What cell markers should we look at for the adult neural stem cell?
Yeah. So I think it's pretty straightforward to count the number of adult neural stem cells, like using the markers and also the newborn neurons, like the double coating, all these things.
Okay.
Yeah.
Great. Thanks. I'll talk more. You will find something interesting.
Yes.
Thanks.
Yes. Thank you.
Now, open questions for first Hongyan, please.
Yeah, Yukiko, really nice talk as usual. So I have a question is that, so it showed at last pretty quickly that you think BMP4 is BMP signaling is probably important for regulating the P57 expression in the embryonic stages. So because BMP signaling is also important for estroglycogenesis. So how does the cell, decide whether they becoming slow dividing neural stem cells or they're becoming estroglycan precursors?
Yeah, that's a very, very great question. Thank you very much. So we think that the BMP signaling alone is not sufficient to establish the adult neural stem cell lineage. So the combination between BMP and NOT signaling is very important. So in this case, so BMP treatment itself is so not just in the in vitro but also in vivo. So the persistent NOT signaling via HEY1 that is not just induced by the so the combination of the NOT signaling and BMP is necessary for inducing the HEY1 at the sufficient level. So I think that these two things are important.
Thank you.
Thank you.
Hideyuki.
Okay, wonderful talk as always. So, okay, I have a question about the behavior of the adult neural stem cells. Okay, so in my understanding, so quiescence is different from the post-mitotic. And let me ask you about trigger that activate quiescent neural stem cells. I believe that ASK1 regulation plays important roles. Maybe HES1-mediated repression, if it is removed, it activates neural stem cells. That is one thing. Is it then how it's regulated? Okay, that's my question. The other is that choroid plexus has produced a lot of factors. So some years ago, Chris Walsh in Children's Hospital reported mitotic activity is induced by the choroid plexus derived factors. How do you correlate the BMP's actions? That's my questions.
Thank you. Both are great questions. So the first one is how are the adult neural stem cells activated? So we actually examine the level of 57 in response to the running the mice, and we saw a degradation of 57 in very efficiently. So so there must be the upstream factors that blocks the stabilization or induce the degradation. We don't know what signal it is, but it's quite likely that AKT is always involved in the activation of these cells, stem cells. So I think it's really possible that AKT goes into that direction. And the second one is the what was that? The choroid plexus and cerebrospinal fluid. Yes. So the Chris Walsh they show that IGF-2 is involved in the proliferation of the neural progenitor cells at the ventricular surface. But I think it's the so IGF-2 and BMP are secreted factors. So basically, all the cells are exposed to these ligands. But we think that the BMP signaling are somehow that starting with all cells may be exposed to BMP and IGF, but there may be a segregation the process after the BMP and IGF signaling. So, and that discrimination probably involves the notch lateral inhibition, and also we think that the BMP signaling induces the BMP antagonist downstream of this signaling. So it's possible that that's also involved in this discrimination of the neural stem cells and surrounding cells.
Okay, receptor or regulator level will be very important.
Downstream the BMP, the receptor, that there might be the signaling that discriminates the BMP positive cell versus BMP negative cells in the later step.
Okay, thank you. Thank you very much.
Thank you for the great questions.
Stuart, please. Oh, sorry.
First of all, Yuki, so nice to see you after so many years. What a great talk.
Yes.
Great to see you. So, this is kind of a corollary of the last talk. Do you think there's a potential for therapeutic conversion of adult neural stem cells by inhibiting the Notch-A1 signaling pathway after an injury or a degenerative disease? I'm sure you've thought about that.
Right. So it's very, yeah, we really think that so Notch signaling or Hay pathway may be, if you manipulate it, so you might be able to sustain the certain population for a longer period. So that's quite possible. Thank you very much for pointing out.
Makes sense, Stuart. Ian, please. You're muted. I couldn't hear. You're muted.
You're still muted. Thank you. Yes.
Oh, hi, Yukiko.
Hi.
Beautiful talk. I really liked your finding that in the fast cycling neural stem cells, the effector of Notch has 1 has 5, whereas in the slowly dividing neural stem cells, it is the Hay1. So my question is, how this kind of effector selection is controlled?
Yeah, that's again really great question. So yes, so why these effectors are selectively activated under the same Notch pathway, right? So the Michael Elvis group, they published the paper in 2018 that this sustained Notch activation can actually induce the Hay expression, but the transient, the Notch activation can induce the Hess induction. The mechanism is not known, but I think that's one of the reasons that you can see differential activation of not Hay and Hess family members. And in that case, so actually in his paper, they show that the activation of Notch is also so the transient activation of Notch is induced by delta-like ligand, and the sustained activation at delta-like 1, but the sustained activation of Notch is induced by delta-like 4. So I think that the kinetics of Notch activation is important, is the key for the induction of differential effectors.
So that means the ligands also need to switch.
Sorry?
So the ligand, the delta ligand also need to switch when the adult neural stem cell when the quiescent one need to be selected out of the embryonic origin.
Yeah, another great question. So we don't know about the ligands actually. It can be delta-like 1 or jagged 1. But in this case, so as I showed you during the talk, so it's possible that the CDK inhibitors are the ones that is responsible for the stable activation and the stabilization of active Notch, the intracellular Notch. So in that case, the intracellular Notch, active Notch can be it's not transient probably, so it's there for a long time, and that can explain the expression of Hay1 rather than His1 and His5.
Okay, thank you so much. Beautiful work.
Thank you.
Thank you, Kiko. Thanks so much for a beautiful talk and so many questions. Okay, Aaron, so it's starting the Suzanne students talk. Thank you.
Okay, thanks, Zilong. Thanks, Yukiko. Awesome talk. So now it's really an honor to introduce our next speaker, my colleague at Stanford, Dr. Suzanne Pfeffer. And when I first came to Stanford and I was in seminars and she was there, I was a little bit starstruck because she's a legend in the field, and it's wonderful to have a great colleague like her. So Dr. Pfeffer did her undergraduate degree at Berkeley, then PhD in biochemistry at UCSF. And during her PhD, she had important papers where she systematically, using biochemical methods, purified and defined the components of clathrin-coated vesicles. She then did a postdoctoral fellowship with Jim Rothman at Stanford, and she continued to study vesicles, but now looking at transport of how mechanisms of transport of proteins from ER to Golgi and within the Golgi. And then she launched her own laboratory at Stanford in the Department of Biochemistry and studied transport of proteins within Golgi, always using biochemistry, defining the components of the process that she was studying, reconstituting them. She had major findings about using biochemical analyses of Rab GTPases, how those worked. And given her seminal findings and her expertise, it really led into her being the key person in the field to help figure out how this Parkinson's disease gene, this most commonly mutated Parkinson's disease gene, LRRK2 works, when it was appreciated that one of its most important substrates and targets is a Rab GTPase. She's been she's an elected fellow of the American Academy of Arts and Sciences. She's an elected fellow of the American Society for Cell Biology. She served as the president of the American Society for Cell Biology. At Stanford, she was the chairman of biochemistry department, and all around an inspiring scientist and rigorous biochemist. And looking forward to hearing your talk, Suzanne.
Thank you so much, Aaron, and to both you and Zilong. Thank you for organizing this symposium series. What a wonderful opportunity that you guys have catalyzed to bring everybody together. So as Aaron told you, my work has turned from Rab GTPases to understanding the molecular basis of inherited Parkinson's disease. Today, I'm going to focus on our work in brain. And I have to say, my PhD was on synaptic vesicle protein recycling in Clathrin-coated vesicles. So coming back to brain is a big circle over many, many years. And Lou Reichard, who's on the call, was on my thesis committee. So thanks for joining us today, Lou. So I'm going to share my screen. Okay. So today, I want to tell you about our work on Parkinson's due to mutation in leucine-rich repeat kinase 2. And I'm going to tell you how it links to hedgehog signaling. And I just want to point out that as we study the effects of LRRK2 pathogenic mutations in this kinase, we want to focus our understanding on how it specifically gives us Parkinson's disease and not Alzheimer's or some other form of neurodegeneration. So in our thinking, we have to turn to always have in mind the fact that Parkinson's disease is first and foremost the loss of dopaminergic neurons in the substantia nigra. So everything we think about as part of my presentation has to come back to those specific neurons. And also I want to point out that our work is funded by a program called Aligning Science Across Parkinson's Disease, which encourages open science and team collaboration. And because I'm new to this field, I really welcome your feedback, and I'm going to give you a progress report today on our work. This is our most important collaborator, Dr. Dario Alessi. He's the director of the MRC labs of protein phosphorylation and ubiquitination in Dundee, Scotland, perhaps the most generous collaborator I could imagine. This is my team, and today a particular shout out to Herschel Dekne and Shahzad Khan, and I will point out their work as I go along. So the leucine-rich repeat kinase is a gigantic protein. It's a very unusual protein, 2,500 amino acids. It may function as a tetramer, sorry. It has a protein kinase, but unlike most other kinases, right next to the kinase domain, it has a small GTP binding domain that looks structurally just like a Rab GTPase. And that should have been a clue a long time ago. But what's also unusual about this protein, it has to bind GTP for the kinase to work. And that's almost unheard of. And about 6 years ago, my collaborators, Dario Alessi in Dundee, Matthias Mann in Martinsried, Germany, discovered that among the most important substrates of this kinase are the small Rab GTPases that are master regulators of membrane trafficking. Now, shown at the top here are particular mutations that cause disease. What I want to point out to you is that they're in two domains, the kinase domain and in the GTP binding domain. And in every case, those mutations increase the level of activity of this enzyme inappropriately. And so our job as biochemists and cell biologists is to understand what's the consequence of that inappropriate activation, and then how does that eventually lead to death of dopaminergic neurons. In a lot of work I'm not going to have a chance to tell you about, and really predicated on insight from Dario Alessi. We know that Rab phosphorylation is predicted to interfere with the ability of Rabs to bind to their partner proteins. The phosphate is right on the side of the molecule that would be critical for those interactions. Now, importantly, what happens is when you phosphorylate a subset of Rabs, it completely flips a switch. So instead of binding their normal partners that are important for their normal roles, you get enhanced binding to a new set of partner proteins. And it's these interactions with proteins with very funny names that have dominant effects on cell physiology. Of the 65 human Rabs, there are about 10 that are true physiological substrates of this kinase, but Rab10 is perhaps the most significant together with Rab8, and it is complexes of phosphorylated Rab10 with proteins, especially RILP-L1, that have dominant consequences in cells. Now, if this were happening on a large scale, it would be deadly for cells because they need Rabs to carry out normal protein secretion and organization of the secretory and endocytic pathways. But it turns out at steady state, only a very small percentage of Rab proteins are phosphorylated at any time and forming these complexes. Nevertheless, they do have dominant phenotypes that we need to be aware of. And all of this can be reversed by a phosphatase, first also discovered by Dario Alessi, called PPM1H. And we've helped to characterize the cell biology of that particular enzyme that seems to be a phospho-Rab specific phosphatase. And I won't say much more about that, except we have a mouse knockout and I'll tell you about the phenotype. Okay. So Herschel Deckner in my lab, Martin Steger in Matthias Mann's lab at the same time discovered that primary cilia formation is blocked when cells express pathogenic mutants of the LRRK2 kinase. And this is an example of cultured 3T3 cells. You see the nuclei stained in blue. You see primary cilia stained with an antibody to RL13B. And this green cell is expressing pathogenic LRRK2. And I think everyone can see it is not ciliated. And if we simply add a small molecule inhibitor of the kinase to these cultures, despite the presence of pathogenic LRRK2, we see a very nice primary cilia. Now, primary cilia are essential for the ability of cells to respond to a hedgehog ligand. Primary cilia emanate from all cells, neurons, astrocytes, except certain cells of the blood lineage. And what's shown here is in the primary cilia, at an arresting condition, the receptor for hedgehog, which is called patched, resides almost exclusively in the primary cilium. In the presence of ligand, what happens is this particular receptor changes the availability of something we call accessible cholesterol, specifically in the primary cilia outer leaflet. It leaves the primary cilium, and the smoothen protein then becomes concentrated in the primary cilia and begins the signaling pathway, which ends in a transcriptional response. And all of this requires primary cilia, and that's very important for what I'll tell you about today. What we showed, this is work again of Herschel Deckner, a postdoc in my lab, is that if we used undifferentiated iPS cells that are patient-derived iPS cells, that we saw that in culture, these cells failed to produce significant cilia. And when this cell line was corrected by TALEN approaches, the cilia were then seen. So this is identical background except for repair of the mutated locus. So we can quantify this and we see about a 50% decrease in cilia. And we can measure the ability of these cells to respond to hedgehog by looking at downstream expression of GLI. And we see that the response to hedgehog, as monitored by GLI expression, absolutely mirrors the presence of cilia. This is a classic paper from the work of Takeshi Kaneko that many of you are familiar with, but for the non-experts in this area, it really gives you a good sense of the anatomy of the system that we're studying. What Kaneko and colleagues did was use a Synbis virus stereotactic injection directly into the substantia nigra of a virus expressing palmitoylated GFP to enable tracking of the projections from single neurons, dopaminergic neurons emanating in the substantia nigra. So what's shown in blue is the pathway, if you can follow my cursor, of a single neuron coming from the substantia nigra here all the way into the striatum, right here. And I'll show this blown up in the next slide. It's quite remarkable. These are the projections in blue of a single neuron emanating from the substantia nigra. And so these are all communicating with neurons and astrocytes in the dorsal striatum. And those are the ones we're going to focus on. Our work is really influenced by this paper from Andreas Kottmann and colleagues from the City University of New York. They were studying sonic hedgehog in relation to this same pathway that I'm showing you. So down here are the dopaminergic neurons of the substantia nigra. Up here is the dorsal striatum. In this study, what Kottmann showed is that these neurons, the tyrosine hydroxylase positive neurons, require sonic hedgehog for their survival, but they do not have sonic hedgehog receptors. Instead, they secrete sonic hedgehog into the dorsal striatum, and he also showed in this paper that the cholinergic interneurons, these are 2 to 3% of the total neurons in this region of the brain, are also dependent on this sonic hedgehog for their survival. And in exchange, upon receipt of that signal, they send back GDNF as a neuroprotective factor for the dopaminergic neurons. Because this is focusing on the nigrostriatal circuit and is directly relevant to cilia, we wanted to find out what was going on with the ability of these cholinergic neurons to sense this sonic hedgehog signaling, because we had seen a ciliary dependence in cell culture. And this is work from Herschel Deckner from my lab. What he showed is that in this region of the brain, it is exactly these cholinergic neurons, and only these cholinergic neurons, that lose their primary cilia in mouse models of LRRK2 Parkinson's. And I'll show you that data next. So this is from the dorsal striatum of a mouse carrying an R1441C LRRK2 mutation. The cholinergic interneurons are stained with a cholinacetyltransferase antibody. They're shown in green. The cilia on these neurons are labeled with adenylate cyclase 3 antibody, and you see a very nice cilium coming right from this nucleus region of this cholinergic neuron in the wild-type animal. The surrounding nuclei are from medium spiny neurons that are the predominant neurons in this region of the brain. They're perfectly nicely ciliated. And when we go to the mutant animal, here's a cholinergic neuron. You see they are lacking cilia very clearly. And we can quantify that, And what Herschel showed is that, this is too far back. If you quantify the medium spiny neuron cilia, they are really unchanged. Those are shown in the dark bars, but the gray bars show you that the R1441C mutant animals have about 50% the level of cilia as normal in the mutant animals compared to the wild type. So what you would predict from this is that these cells would be less able to sense sonic hedgehog. They would be more vulnerable to loss of this essential growth factor for them. And you would expect also that they would decrease their ability to send back neuroprotection to the substantia nigra. And it's important to remember, Parkinson's is a disease of aging. So it may be a slow insult over years of human life, months of mouse life, that could yield significant death in that region. This postdoc, Shahzad Khan, has gone on to show that it's not only in that model of mouse, but it's in 2 other mouse models with different mutations in the LRRK2 protein that have identical phenotype. And most remarkably, in the knockout mouse that doesn't have the phosphatase, so it will have inappropriate levels of hyperphosphorylated RAB, we see the identical phenotype, loss of primary cilia on cholinergic interneurons in the dorsal striatum. What Shahzad also discovered is that uniformly astrocytes in the same region have also a cilia defect. And now I'm going to show you some very recent results. These are from human patient samples from the Banner Brain Institute in Arizona, We're looking at astrocytes in the striatum. These are control astrocytes. You can see cilia shown here in red. We stain these cilia with RL13B. We have analyzed sporadic Parkinson's patients and 5 G2019S carriers, which are age controlled in our control samples, are of similar age. And we see a complete deficit, or almost complete deficit, in primary cilia in this region of the brain for these astrocytes. And when we go looking for the cholinergic interneurons, we aren't even able to find them preliminarily in the brain, this brain region, for sporadic patients or for patients with LRRK2 mutation. So it's clear there's a very serious defect in hedgehog signaling to non-existent cholinergic neurons that are controlling this neuroprotective pathway in human disease, which we are, of course, interested in continuing to study. Okay. Yukiko Sobu, a former postdoc in the lab, did single-cell RNA-scope in situ hybridization experiments. What she found is, here we're again back in the dorsal striatum of the mouse. Here you can see this is a R14. This is a wild-type cholinergic interneuron, nicely ciliated, and she could look specifically at the level of hedgehog signaling in those single cells using a probe for GLI1 transcription. In the cells that are remaining that have cilia, even though there's a cilia loss in the LRRK2 mutant, this is a ciliated cell in the mutant cells, she saw an overshoot. In other words, these cells were inappropriately overexpressing the GLI1 downstream reporter, as if they either had too much ligand or too much stress from the fact that there weren't other cells around to present or provide neuroprotection downstream. Now, the story sounds good, but we really need to see what's happening with that neuroprotective factor. And this is a very recent result from Shahzad Khan. This is from single nuclear RNA-seq that we've done specifically of the dorsal striatum, comparing only, we're looking specifically at the cat neuron transcription profile from that region of the brain by single nucleus RNA-seq. And you can see highly significant lower levels of GDNF RNA in the cholinergic interneurons when they have a LRRK2 mutation compared to the wild type. So this really brings us back to this model that's also true in Parkinson's, where this, at least in the inherited Parkinson's and possibly in the human case that we've been studying, these cholinergic neurons are playing a really important role. There's a decrease in their ability to synthesize GDNF because of a cilia defect that's caused by LRRK2, and that will give less neuroprotection to the tyrosine hydroxylase positive dopaminergic neurons of the substantia nigra. So we have some unanswered questions that we really are hoping that the single nucleus RNA-seq are going to give us some clues to. Why is it that only certain neurons lose cilia in the brain? I told you that all those medium spiny neurons have perfectly normal cilia. So there's something special. It may relate to which cells make more of the phosphatase to protect from inappropriate kinase activity, and we'll have that answer soon. What do neuronal cilia do in the adult brain to modulate neuronal function? What we know is G protein coupled receptors, neuropeptide receptors, neurotrophic factor receptors are concentrated in the ciliar membrane itself. So it's not just hedgehog signaling that will change in the presence of a defect in cilia. Other things will change, the ability of these cells to talk to each other. And of course, we'd also like to understand what the consequence of astrocyte cilia loss is on the overall circuit. What are those cilia doing to support the synapses in the dorsal striatum? So I want to just give you some hints, again, work in progress for this specific region of the mouse brain. We're busy cataloging the different cell subtypes. For example, in this region of the brain, GFAP is not a good marker of astrocytes. S100 beta is, because only one subclass of astrocytes in this region of the brain are GFAP positive. But they all are positive for other astrocyte markers. And so we're beginning to subcategorize. This could be as many as 9. It may be as few as 2 different types of astrocytes in the brain, and we're busy validating the markers for this. but we already have some interesting results that I can share. For example, all the astrocytes are responding to hedgehog ligand in both the wild type and the mutant case. They all have glutamate receptors, as you'd expect. But interestingly, you'll see about half of them have LRRK2 in both the mutant and the wild type animals, and half of them really don't express much LRRK2. And that's completely novel observation. People have assumed that, including starting with Ben Barris's RNA-seq analysis, that this was a highly expressed astrocyte marker. But in fact, there are subclasses, and we want to understand, are these cells, the astrocytes that express more LRRK2, are they more vulnerable to the mutation? Are they the cells we're looking at that are specifically losing their cilia? And it's these kinds of classifications we will continue to carry out. And then finally, I want to give a shout out to this amazing story from Suzanne working with David Clapham, and this will be coming out next week in Cell. What he has seen by incredible morphological approaches is synapses that are functional directly onto primary cilia. It's the most amazing paper. If you haven't seen it, it's in bioRxiv right now. And we are very excited because we would like to look for these kinds of synapses to understand what these ciliary deficits are doing for LRRK2 patients and our LRRK2 mice. And with that, I will stop sharing my screen and open my talk to questions. And again, thank you so much for giving me a chance to share our ongoing work.
Thanks so much, Suzanne. Amazing talk. Xuecai, first question.
Thanks. Hey, Suzanne, really nice work. And I learned a lot. I was a former postdoc of Matthew Scott.
Ah, thank you.
Yeah, so really I learned a lot about from your talk about hedgehog signaling and Parkinson's disease. So a quick question I have is, a lot of mutations are familial mutations. So embryo has it too. So do you have any, this is more like speculation type of question, maybe those mutations might impact development of the brain?
So what you notice, in no case do you lose all the cilia. There's always some cilia there. And so many people have said, is this a ciliopathy when you have a mutation in LRRK2? It's not, it is a ciliopathy in that you have an effect, but obviously for some reason, cells are, some cell types in the animal are completely okay. And so there's no question that primary cilia are needed for development. They're needed for brain development. You can knock out primary cilia and have all at different stages of development, and also even in the adult, and have different phenotypes. But I think it's because you don't have full loss of cilia that you're able to develop, and the brain seems to function pretty normally. For us to see the phenotypes, we only need to look at it. We see our phenotypes at 8 weeks of age in the mouse. So it's not that you have that the cilia are lost at late stages. And as you're also familiar, in the mouse, we know that the cilia get longer with age. So they are changing almost as if they have to get longer because they're doing less good a job of sensing and signaling. So it's an interesting question about how do the mice develop so well with a cilia defect. And I think it's just because it's limited in terms of which cells are affected.
Thank you.
But you bring up another really important point, which is that every cell we've looked at in cell culture, including primary astrocytes and other cell types, show a cilia defect. But when we look in the animal, we can't recapitulate that. In the animal, for example, the medium spiny neurons are totally normal. So we think that will be a challenge if we wanted to use, for example, neuronal stem cells in culture, or astrocyte stem cells in culture, that they may not do what they do inside the brain.
Thanks.
And Xuecai has an amazing, still unpublished story about proteome of cilia in vivo in brain, which could well be of interest. Hope to be published. Yeah. Okay. Yeah. So Hideyuki, you have a question?
Okay, so great talk. And so it's very interesting that only subset of neuron has loss of the cilia phenotype. So recently, based on spatial transcriptome analysis, Evan Markowski's lab in Broad Institute have shown that only ventral half of the substance nigra is very vulnerable in Parkinson's disease. Do you think that there's like a difference in the sonic hedgehog expression pattern and the substance nigra, and do they contribute some neuron-specific phenotype in the striatum?
I think there's going to be many different kinds of cells communicating in many more specialized ways than we've got so far. So we've only mapped out the responses in the dorsal striatum, and one's going to need to do the same kind of single nuclear analysis in the substantia nigra, and that is in progress, and we will have much more information soon. But yes, I mean, because there's the neurons are going to different places and also getting feedback from different places, and so we need to match those. And I think that's a really interesting question.
Okay, thank you.
Yes.
Hong Yan?
Oh yeah, Suzanne, nice talk. So question from non-expert. Have you tried to supplement or like enhance the level, just adding back GDNF or enhance the function of cilia so that you can rescue the phenotype by the LUK2 mutation?
Couple of things there. There are LIKE2 kinase inhibitors that are brain penetrant that are currently in clinical trials. And the question is, would that be enough to bring back cilia in the brain? So we fed animals for 2 weeks with this drug with the hopes of bringing back cilia. We were not successful at bringing back cilia in this region of the brain, even though we have evidence that the drug did get into that part of the brain, or at least into the whole brain. We didn't dissect substantia nigra to detect the drug there. A challenge, and of course we hope that you will be able to reverse this effect if that drug's if this is going to help patients, if this is the mechanism by which one of the ways it's helping patients, we hope those cilia will grow. But the challenge, of course, is, and this relates to Yukiko's talk, cilia are very cell cycle dependent structures. And neurons are not non-dividing cells. And so the question is the blockade one that can be reversed, or do you need a cell cycle to grow a new cilia? And so I'm hoping that it will be that we can reverse that effect. With regard to the ability to use GDNF to help patients, there were trials some time ago using GDNF that were not entirely successful because you probably also need the hedgehog signaling. But there's a recent paper from my colleague Andreas Kottmann who set us onto this whole circuit, which is really interesting. So it turns out one of the few ways you can help patients with Parkinson's disease is you give them L-DOPA and their tremors will immediately stop. But after a few years of L-DOPA, they acquire a condition which is called L-DOPA-induced dyskinesia. And this is a form of movement with very jerky motions, and it's not very pleasant. And patients often refuse L-DOPA treatment because they don't want the dyskinesia side effect that will come with time. And what Andreas Kottmann has shown is if you use hedgehog modulators in conjunction with L-DOPA, you actually can rescue this dyskinesia effect by balancing out the hedgehog signaling on both sides. So by using hedgehog agonists in conjunction with GDNF, it may be of great benefit to patients, especially getting rid of the dyskinesia. And so we're very interested in following that up. And he's seen that in 3 different mouse models, rodent models of Parkinson's, and also in macaques. It's very compelling data.
Thank you so much.
So, Suzanne, maybe a little bit more of a provocative question. You said that you treated mice with the LRRK2 inhibitor, that you fed them it.
Yes.
What if that is telling us something important? Maybe there's some other effects other than just the kinase role, or maybe there's even a loss of function that might be contributing to some of these phenotypes. I'm just wondering if you've looked at the knockout animals or, knockdown situation, if the LRRK2 gene is required for.
So what we know yes, absolutely. So the knockout animals, so type 2 pneumocytes in your lung, the same cells that are targets of CoV2, those cells need LRRK2 to secrete surfactant. And if you accumulate surfactant containing granules in those rare cells of the lung, if you have no LRRK2 at all. But imagine, for a LRRK2 inhibitor to work, you don't need to inhibit it all. The mutations increase kinase activity twofold. So presumably you just need to inhibit half as much, and that might be a therapeutic regimen that could work. Denali people, they're in clinical trials, think you have to go a little bit higher, but I don't think you have to inhibit it all. But the mice are otherwise pretty okay. So
No, but you're postulating that the kinase inhibitor is not, or you're saying
I'm saying
the kinase
2 weeks
inhibitor
is
doesn't.
not enough to regrow a cilia in the brain, because in general, we don't know what it is you're reversing. And remember that, sorry.
Does it reverse the RAB10 phosphorylation though?
It does reverse the RAB10 phosphorylation, but it's complicated because there's a lot of the phosphatase there. It's a complicated answer.
I'm just saying,
yeah.
what if the cilia and the hedgehog defect is not due to a gain of function in LRRK2? What if it's right? That's all I'm saying.
So again, the full cause of Parkinson's we need to figure out. The inhibitor does work in terms of changing secreted lipid levels due to kidney secretion. That's a LRRK2 sensitive thing that you can easily measure. I think the changes in the brain are hard, but separate from LRRK2, Sarah Getz at Duke University, who trained with Catherine Anderson, and she's a world's expert on cilia, she showed if you block cilia by genetic means, and asked, can the cilia come back? She has to wait months for them to come back in the brain.
Okay.
I think that's a better answer. So sure. I'm hopeful that longer term LRRK2 inhibitor treatment could be of benefit to patients.
Got it. Okay. Any other questions? No. Okay. Suzanne, thanks so much. Really amazing.
Thank you very much. Thanks, everyone.
Yeah, thanks, Yukiko. Thanks, so long. Thanks, everyone. See everyone next week.